Hey everyone, this video is for the lab for week 4. We're going to talk about how you count and analyze microbial populations. Okay, so I'm going to share with you this PowerPoint for the lab. Now this is all material from section 6.4. Let me just make sure 6.4 in your textbook. Okay, so when we talk about measuring microbial populations and population growth, there's a couple of different ways that we can do it. One way, of course, is we can count them directly, but this is a challenge given that these are microscopic organisms, so we need to count them in a microscope. There is a special kind of slide that we can use called a Petroff-Hauser chamber or a cytometer. These aren't used very often with bacteria, but if you have decent sized bacteria, it's possible. So essentially what you have here is you have a defined volume. So the depth of the, this is a wet mount, so this works best with a phase contrast scope so that you can see these live cells. These aren't stained cells, right? So this would be like a drop of a broth culture, say that you wanted to count the density of cells in. So you would put it on this slide. This slide has etched into it a grid where the grid markers are a known distance apart. And then when you put the glass slide down on top of it, the glass slide is also held a specific distance above the slide itself, so that you have essentially a three-dimensional grid with known, so you have a known volume that you're counting is the bottom line. Okay, and then you can count how many cells are within that grid, and you can actually you can then calculate the density of cells in that grid. The idea is that it's cheap and fast and easy, although in reality it's not really any of those things, but in theory, that's what it is. I should say with a flow cytometer and larger cells, you can do this easy. A computer can just flow particles through and can calculate the number in a given volume. But bacteria are typically near the lower limit that you can use a flow cytometer for. So it's a challenge with bacteria. Another big problem is that you can't differentiate living cells versus dead cells. And if that matters to you, and it often does, then all you're seeing is essentially cells there, so you don't know whether they're alive or dead. All right, so direct counting with bacteria is a challenge. So another way you can do it is an indirect method, and that's what we would be doing in the lab, or that's one of the things you would be doing in the lab this week. So you're going to do it in this lab worksheet and exercise in this. So this is called viable cell counting. So if we do a dilution series and put them on a plate, put a sample of each dilution on a plate and see how many colonies grow, then we can extrapolate that back to how many colony forming units, or CFUs, were there in the initial culture. So what that would look like is you would take our diluted samples, and we don't use the pour plate method very often, although it does have some advantages over the spread plate. But if you remember early on when I showed you what was in your lab bench, there was this little triangular metal rod in there. And so that's something you can use to spread a sample evenly out on a plate, and then you can look at how many colonies on the plate are on the plate. The advantage of the pour plate method is that the colonies get embedded in soft agar, so they don't grow as much, so you can get more on a plate and still count them accurately. If you have an organism that likes to form big colonies, then then you might not be able to get as many on a plate, or you just don't incubate them for as long. So spread plating is a very common method for measuring viable cell density. So you do a dilution series, you plate a known amount, in this case, 0.1 mils on each plate, and then you calculate it. So this is an example of what that kind of data would look like right here, for example. So at the 1 hour time point, we did no dilution, we did a 10-fold dilution, 100-fold dilution, and 1,000-fold dilution. So what that would look like in practical terms is, say, we would have our starting culture, we would plate 0.1 mil of that, see how many colonies we get. We'd take our starting culture and start diluting it. So we'd take 0.1 mils, add it to 0.9 mils of sterile media. That would be our 1 to 10. Then we'd take 0.1 mils of that, add it to another 0.9 mils of sterile media. That would be our 1 to 100, and onward, right? The data that is on your worksheet actually doesn't even show you the plating values for these, because the bacterial cell density is a lot higher to start with. So it starts with actually 10 to the minus 4th and goes to 10 to the minus 7th, I think, right? And so then we have the number of colonies. Replicate plates just means you did 2 identical plates, each of them with 0.1 mils, and then you calculate the CFU per mils, okay? Now I realize that you haven't, we haven't talked about how to calculate CFU per mil from this kind of data, so I'm going to stop this for just a second and show you on the board how that would work. So the CFU per mil value would be equal to the number of colonies on the plate, right? So that, you know, could be anywhere from 0 to thousands, but we're only going to use plates with 1 to say 300 colonies for this calculation. And the reason for that is that if you have too many colonies on a plate, then you can't count them accurately, because the plates, the colonies start to overlap with each other, okay? So we're going to limit the number of colonies that we're going to say is a useful plate to keep it, you know, relatively low. Okay, so the number of colonies on the plate divided by the volume in milliliters, right? Because this is our denominator, milliliters here. So remember, in the example on that slide, they only plated 0.1 milliliters, right? And in the example in the worksheet, they only plated 0.1 milliliters, right? So we have to take that into account, because if we actually had plated a whole milliliter, it would have been 10 times more colonies, right? So that's in our denominator here. And then here is the dilution, right? So if there was no dilution, right, then this would just be, well, for our purposes, it would be 1. If there was a 10-fold dilution, then it would be 1 times 10 to the minus 1. If it was a 100-fold dilution, 1 times 10 to the minus 2, etc., right? Okay, so this is how you're going to calculate CFU per mil, right? Go back to sharing our screen now. Let's go back to that PowerPoint slide. Okay, right? So in this calculation here, say for this 1 to 100 dilution, right? 83 colonies, that would be on the top of your calculation, right? And then on the bottom, on the denominator of the calculation, would be the volume, which is 0.1 mils, and the dilution, which is 1 times 10 to the minus 2, right? This way of expressing it doesn't do us any good mathematically. We have to express it like this, okay? And that's how we're going to get to our number, right? That's how you calculate CFU per mil. Now this down here says too few colonies to be meaningful, less than 10. I just said in the lab worksheet, anything greater than 0 and less than 300, okay? So if it's 0, don't use that in calculating the CFU per mil. There's nothing there, okay? So at any rate, it says now try it out. What are the CFU per mil values at 1, 2, and 3 hours? Calculated using the numbers listed here. That's actually all shown there already. But again, you're going to be doing this in your worksheet, okay? Oh, I just realized it didn't, yeah. Anyway, all the numbers. So I just asked you to calculate it, but the numbers are already shown there, okay? So make sure that you can do that calculation of CFU per mil correctly, right? Okay, here's a question for you. What if the cells are already very diluted? Or essentially, what if the concentration of cells in your sample is less than like 1 per mil, right? And this might be if you were doing like environmental water sampling or something, and it was fairly clean water. Well, you can then use a filter apparatus to concentrate the cells so that you can use larger volumes than you can easily plate on a plate. Really, you can't put more than about 100 microliters on a plate or it just gets too sloppy, right? So if your cell concentration is relatively low, then you might want to catch them on filters and then put that on a plate, right? Or concentrate it that way. Okay, the other thing to talk about is that, you know, there's another way of doing estimating cell density in a broth culture, a liquid culture, and that's to use turbidity, essentially in a spectrophotometer. So when the spectrophotometer tries to pass a light beam through a liquid sample, the more bacteria there are, the more the light will be scattered and the less of it will make it through to the detector. Okay, so that is a way of looking at either absorbance of the sample or with particulates where it's not really absorbing, it's scattering light. We refer to that as the optical density, which is usually abbreviated OD. Okay, and furthermore, we usually pick a particular wavelength to use to do this measurement. And so for bacteria, it's usually something like 550 to 600 nanometers. So in the lab worksheet, the OD is going to be measured at 600 nanometers. Okay, so OD 600 is optical density at 600 nanometers, and that's a measure of the turbidity, which is the density of cells in the sample based on light scattering. Okay. Now, the important thing here is to talk about the idea of a growth curve, because that's what is being looked at in the lab this week. Okay, so a growth curve is looking at growth of a microbial population in a closed system, a batch culture, right? So a batch culture is basically you start with a flask of media, you inoculate the cells in it, you cover it up. So the nutrients that the culture has available is just what's there at the start. There's no addition of new nutrients. Now there is oxygen available. So if they're growing by respiration, we're doing this in a shaking culture, so they're not limited for oxygen, right? But in terms of carbon, nitrogen, and that sort of thing, for incorporating into biomass, the amount of nutrients that are there at the start is basically what they have accessible for. Right. So essentially, we get what's called logistic growth, which is that during the exponential phase, one cell divides into 2 cells. Those 2 cells then divide into 4 cells. Those 4 cells divide into 8 cells. Okay. So if you were plotting this on a linear scale, you would have an upward sloping curve, right? But if you plot it on a logarithmic scale, this exponential growth now looks like a straight line, okay? So our microbial growth curve is going to be a semi-log plot, meaning one of the axes is a log axis, that's the y-axis. The other axis is a linear axis. Okay, now I just realized this is a little misleading because I don't want you to plot your samples the way it's shown here. In this one, it looks like a straight line because they've taken the logarithm of the value of the population density and plotted that. I want you to actually plot your numbers directly, but set your y-axis to a log scale. Okay, I'll talk about that in lab, but I just want you to make that clear. At any rate, for purposes of this illustration, it doesn't matter what we have as a log scale here and a linear scale here. Okay, now you notice that the logarithmic growth doesn't start immediately, right? Typically at the start of any given experiment, we have what's called a lag phase, because often we are taking a sample that's not growing happily. We might be taking a sample from a colony on a plate. We might have used a starter culture that was grown in a different media or has been sitting there on the bench or something. So often there's a period of adjustment. That's the lag phase. The growth rate during lag phase is lower than it is during the log phase or exponential phase. Okay, so the log phase or exponential phase is defined as having a constant growth rate, which yields a straight line on this log scale. In the lag phase, the growth rate may go from 0, so essentially there's no net growth, to an accelerating growth rate until it reaches its maximum growth rate, which is the log phase, okay? So during log phase, we have a constant growth rate until the culture starts to exhaust its carrying capacity, starts to exhaust nutrients or nutrients accumulate. Now, shown here is a stationary phase where there is no net growth. It's important to recognize there's also a transition from the true log phase, where growth rate is constant, into the true stationary phase, where growth rate is 0, okay? So this is what we call a transition phase. And I want you to recognize that this is not truly part of the exponential or logarithmic growth phase. Growth is slowing down here. So each generation, the growth rate is getting slower until it stops, right? Now, in stationary phase, the cells are either limited for some nutrient, or some waste product has accumulated. Sometimes the pH has been acidified or even gone more alkaline, depending on metabolic waste products that are being produced. But whatever it is, there is no net growth. Now, some cells might be dividing, but those cells that are dividing are equally matched by cells that are dying. Now, eventually you get, and notice there's a break here because we don't know how long this is, eventually you get to a point where death of cells outweighs any division that's happening. And then we actually start into an exponential death phase, okay? Now there are two different curves shown here, because one of them is based on turbidity and one of them is based on viable cell density. Sometimes those don't exactly track, which is why these aren't shown as being exactly equal. And that is basically because in order for them to exactly track, then cells would all have to be the same size and they would all have to be viable, okay? You notice here that in death phase, the optical density doesn't drop as fast as the viability. That's because a cell can be intact and scattering light, but not alive, okay? So again, there are reasons why these don't exactly track sometimes, but usually they resemble each other fairly well until you get to the death phase, and then they can diverge, okay? So 4 phases: lag phase, log phase or exponential phase, stationary phase, and death phase, with transitions in between them, okay? All right, you may not always see death phase because you may not let your culture go long enough to see it. Lag phase is not always going to be the same, okay? Exponential phase, the rate of growth may depend on environmental conditions. Okay, we're going to talk about that in the class. So all these things can be different for even the same species under different growth conditions, certainly can be different for different species under different growth conditions, okay? And the growth conditions will largely determine the length of each phase and the growth rates and death rates and the length of the stationary phase, okay? So this is just a summary. During lag phase, microbes are getting ready to grow. They're adapting their gene expression, their metabolism to grow. Log phase, they're growing at a constant rate as fast as they can under the conditions you're growing them under. And if that growth rate is constant for many generations, then you see a straight line on a semi-log plot where the population density is on a logarithmic scale, okay? Stationary phase, the culture has reached its capacity for whatever reason, and there's no net growth. And then in death phase, again, the cells actually start to die. They can't stay in stationary phase forever, okay? So from this kind of data, you can calculate generation time, you can calculate growth rate, and you can calculate growth yield, okay? Now here's a key part, and I realize that this video is getting a little long, but we need to just go through these equations at least briefly. For microbial cells undergoing binary fission, where one cell is dividing into two cells, we can calculate a population at a subsequent time point if we know the starting cell density and then the number of generations that have elapsed since then. Okay, so n sum 0, our starting cell density, times 2 to the n, where n is the number of generations. We can calculate the number of generations by transforming this equation if we actually have the cell density or the OD, which we're going to take as being similar to the cell density. We can fit them into these equations the same way. So if we have the cell density at one time point versus the cell density at our starting time point, then the number of generations is equal to 3.32 times the difference between the log of the later time point minus the log of the early time point, right? So that's how we can calculate the number of generations, and that's really an important number to be able to calculate. Now we can calculate the generation time, which is the time that it takes for a population to double if we have two different population sizes. So if we have the population density at two different time points, again, it can be CFU per mil values, it can be OD values, okay? So GE, the generation time, is equal to the elapsed time divided by the number of generations, right? So elapsed time is the difference in time between the two times that we measured cell density, okay? So we may not know the number of generations, but if we know the two population densities, then we can calculate that, okay? So again, that can be in CFU per mil, it can be in OD. And the time can be expressed in hours or in minutes. Often, if you have a bacteria that grows fairly quickly, it makes more sense to express it in minutes rather than hours, right? Now, the fastest growing bacteria have generation times of 10 to 15 minutes, but that's really exceptional. E. coli is actually a pretty fast growing bacteria, and the fastest it can grow is about 20 minutes. It doesn't always grow in double every 20 minutes. In the real world, usually it's, more between 20 and 30 minutes, okay? So just to give you a feeling there. Now we can also calculate another term, which is called the mean growth rate, which is just the inverse of the generation time. So K is expressed as generations per unit time, right? Now for that, it makes often more sense to express the unit of time in hours rather than minutes, okay? So you can calculate it by calculating the generation time first, or you can calculate it from the numbers that we talked about before, okay? So I'm asking you to calculate both G and K for the experiment in your lab this week, okay? Now, when thinking about how those numbers apply to the growth curve, Think about it this way. During lag phase, G, the amount of time that it takes for each generation is actually decreasing. Because if you're not growing at all, the amount of time for each generation is infinite, right? So the growth is the culture is growing, is dividing more rapidly. So G is decreasing, K is increasing, right? During the transition, okay, so during log phase, G and K are constant, right? They're staying the same. And then during the transition to stationary phase, the generation time is likely getting longer and the mean growth rate is getting slower until during stationary phase, again, you're back to where you were. And if you had zero growth at the start of lag phase, which is that G approaches infinity and K is approaching zero. Okay, and then death phase, we're not going to worry about that. Okay, so this is what you should be able to do eventually, and we'll have to talk about this more in class, is to be able to use these values to extrapolate population sizes or to calculate those values if given two population sizes and the time in between them. Okay, I'm not going to go over these practice problems right now. We'll do this in class. Okay. All right. And then the last thing is to consider viruses, right? So we said cells, bacterial cells go from 1 cell to 2 cell, but a lot of viruses or parasites that have invaded a host cell, each generation there between each invasion event, you could actually have one viral particle, remember T4 invading a bacterial cell, and then hundreds of T4 viruses are released at the end. Or parasites like Plasmodium, which causes malaria, invading a red blood cell, and 20 new parasites are released every time that red blood cell lyses. So the equation for population growth looks different now, right? Where the number that you then have the number of generations, it's the exponent, right? The number is different there. Instead of being 2, like in binary fission, it's going to be whatever the burst size is. Okay, and I'm going to stop sharing that for now, and that is going to be the end of this video.